First of all, I'd like to thank the organizers for giving me the opportunity to present my work this afternoon. These are pictures of stones that differ in their shape and color, and these kind of stones are used to build mosaics. If you just look at these stones, all you see is a pile of stones that are of a different color and shape. But once you integrate them into a mosaic, a bigger picture emerges, in this case of Alexander the Great, which is part of a famous mosaic from the 1st century that was found in Pompeii. I actually thought it looked rather nice in this setting. On the other hand, you could argue that without a thorough knowledge of the properties of the individual stones, the artist would not have been able to produce a fantastic piece of art like this. And I think a similar situation applies to studying speciation. We've seen this slide over and over during talks, and it basically shows that speciation is the process where an ancestral lineage splits into two daughter lineages. And if you look at that in a phylogenetic context, each branching event represents a speciation event. You could basically study speciation in two different ways. The first one is to focus on one particular speciation event in great detail and zoom in. And we've seen a lot of examples at this conference so far where people looked at a particular model organism like sticklebacks or the flycatchers this morning. And these kind of approaches are really great to understand the process of speciation. On the other extreme is a more pattern-based analysis. We've also seen a great example of that this morning already, where we can make great use of replication of speciation events if it's studied in a phylogenetic context. And this is the approach that I'll be focusing on today. In particular, I'll be looking at ecological character states at the tips of a phylogenetic tree. Look how these character states change along the branches and how they're associated with speciation. Most of the talks at this conference have focused on animals or insects. Today I'm going to talk about angiosperms, which make up a very significant proportion of ecosystems all around the world. It's a very species-rich group of about 250,000 species, and therefore it's important to understand the processes that drove speciation in angiosperms. One of the most outstanding features about angiosperms is their floral diversity, which has probably co-evolved with insect diversity. And we think that a lot of these floral features represent adaptations to different pollinators. In addition to that, we've got a good conceptual model available, how pollinator-driven speciation could work. And although there's some evidence from lineage or species-specific study, these individual studies really fail to assess whether pollinator-driven speciation is a general mechanism of speciation in angiosperms or not. So how can we test this? Well, if we assume that pollinator-driven speciation equates pollinator shifts on phylogenies, which is an assumption that I'm going to make today, we can simply calculate the frequencies of these shifts. And I performed a phylogenetic meta-analysis to address this question. The first thing I did was a Web of Science search, where in the topic field I entered the search terms phylogen and pollinate. And I got 1,600 hits. Then I took all the studies which presented phylogenetic data as well as pollinator data for the terminal species, which reduced the data set dramatically to only 30 species. In few cases, the pollinators were actually not optimized in these studies, which I then proceeded to do using parsimony and McLeod. The first thing I recorded was the frequency of pollinator shifts. Now, there are really two ways of looking at this. The most intuitive way would be to express the frequency as a proportion of the total number of branches in a tree. In this example, there's a tree with 6 terminal taxa that each have a different pollinator represented by a different color. So this is sort of the most extreme example where you would have a maximum amount of shifts. However, if you reconstruct the number of shifts on this tree, you'll only find a number of shifts which is the number of taxa minus 1. So for the remainder of this talk, I'm not going to express pollinator shifts as a proportion of the total number of branches, which would be 10 in this case, but I'm going to express it as a proportion of the maximum number of shifts that you can reconstruct on the tree, which is the number of taxa minus 1. I was also interested in the amount of convergence in pollinator shifts. So in other words, where the same pollination system would arrive twice. And I used the good old retention index, which I borrowed from cladistics, to express the amount of convergence. The retention index ranges from 0, which is maximum convergence, to 1, which is no convergence. And in this particular example, we've got 3 independent origins of the red character state. The retention index would be 0.33. I scored a couple of clade attributes, which you'll later see I thought are important, such as data source, clade size, taxon sampling, the number of different pollination systems, region and biome that a clade occurs in, and attributes like floral tube or zygomorphy or floral symmetry. And finally, I recorded the direction of a pollinator shift. This is the angiosperm phylogeny, and the arrows represent my sampling, as found in the literature, basically. And as you can see, the early diverging angiosperms are not represented, unfortunately, but otherwise the sampling is spread fairly evenly across the angiosperm phylogeny, although 2 orders are fairly densely sampled, these being the Lamiales and Aspergales, and the latter, obviously, because of the presence of many different orchid studies that are available. The total number of shifts that I found out of a maximum possible of 868 shifts was 32% of branches are characterized by pollinator shifts. And if I average that across the 30 studies, I found an even higher number that included 36.8% of branches were characterized by pollinator shifts on average in these studies, although you can see that the standard deviation is very high, which means that some studies were characterized by very few shifts and other studies were characterized by a very large number of shifts. As far as convergent evolution goes, the retention index was 0.55, which means there was a fair amount of convergent evolution present. Clade size did not have an effect on the proportion of shifts or the retention index, but more importantly, neither did taxon sampling. Why is that so important? So the way I express taxon sampling here is, as you can see on the x-axis, the proportion of extant taxa sampled. Now, if you think about a biological process, extinction, which may bias the result, the signature that's left by extinction would be similar to very poorly sampled lineage, where you basically forget to sample a lot of the extant taxa. Since there's no effect of taxon sampling, I sort of extrapolate that there's probably also not a huge effect of extinction, which is something that we can't really correct for otherwise. The number of pollination systems is positively correlated with the proportion of shifts, which is intuitive. The more pollination systems are available in a particular group, the more frequent or the more pollinator shifts you find. There are a couple of outliers here which I would like to explain. These are 3 clades where each species basically has a different pollination system. Among these are the fig wasps, for instance. So each fig species has a different fig wasp as its pollinator. So therefore the proportion of shifts in these clades is maximal. Most of the data sets that I looked at, 20, had phylogenies that were reconstructed using molecular data, although a few studies combined molecular and morphological data, and there were even some studies based on morphological data only. There's no significant difference in proportion of shifts among these 3 different data types, but interestingly, the retention index is lowest in data sets that include phylogenies that are reconstructed using morphological data. Now keep in mind that a low retention index means a lot of convergent evolution. And this is very counterintuitive, because previously it was thought that data sets or phylogenies that were reconstructed using morphology were prone to circularity, which would lead to very low levels of convergence. And I find basically the opposite result. Probably interesting for those who are familiar with that debate. Regionally, there's a significant difference in the proportion of shifts depending on where the clades occur. I've highlighted this horizontal line here, which represents Australia or Australasia. That's heavily biased towards the 3 clades where each terminal has a different pollination system. These all 3 occur in Australasia. But also you can see that Southern Africa has a very high proportion of pollinator-driven speciation and also a lot of convergence. Although we think that species diversity, both in terms of plants and possibly insects, increases towards the tropics, I don't find an increased level of pollinator-driven speciation if I compare clades that occur in the tropics versus clades that occur outside the tropics. Traditionally, several plant traits have been associated with elevated levels of speciation, and in particular I want to highlight floral symmetry and nectar spurs, where it's thought that zygomorphic flowers are more prone to speciation, as well as plants that possess nectar spurs. And the mechanisms that have been frequently invoked to explain these elevated speciation rates are pollinator-driven speciation. So let's see how these hypotheses hold in the light of my data. I don't find a significant difference in pollinator-driven speciation between clades with zygomorphic versus actinomorphic flowers, basically thereby refuting that hypothesis. And very interestingly, I do find a significant difference in speciation, pollinator-driven speciation between clades with and without a floral tube, but exactly in the opposite direction as was expected and as was predicted from the literature. So I find that clades that don't have a floral tube are characterized by higher levels of pollinator-driven speciation compared to clades with floral tubes. This is probably biased because of the fact that the three clades where each species has a different pollination system don't have floral tubes. The picture that you get if you look at the direction of pollinator shifts is fairly chaotic. You can basically see that shifts are possible in almost all directions. If you look at the top 10 of frequency of shifts, you can see that the most frequent are shifts from one particular bee pollination system to another bee pollination system. And likewise, shifts among different wasp pollination systems and shifts among different moth pollination systems are also very frequent. Shifts from bee to bird are frequent, and they're actually supported by the highest number of studies, largest number of studies. Now, the reason why among wasp or among moth systems shifts are so frequent is probably because the clades that are characterized, where species are characterized by a high number of different pollination systems, are moth and wasp pollinated. Thank you. Bird pollination has often been thought to represent an evolutionary dead end. In other words, once you're bird pollinated, you can't shift away to anything else. My data clearly show that this is not the case. As you can see from the red lines, that there's a lot of shifts away from bird pollination, and in fact a lot of the shifts in the top 10 of most frequent shifts are away from bird pollination. And finally, you can categorize flowers in terms of their tube length according to the different pollination system. So I characterize bee, fly and bat pollinated flowers with short tubes, bird pollinated flowers with intermediate tube lengths, and long-tongued fly, butterfly, and moth pollinated flowers with very long tube lengths. It's been hypothesized that the evolution of nectar spurs is towards progressively longer spurs by the Equilegia study of Whittle and Hodges. Although I do find that the frequency of shifts from short tube systems to long tube systems is higher, I do find an appreciably amount of shifts from long tubes to short tubes. So this is certainly not impossible according to the data that I found. So to conclude, pollinator shifts are very common among plants with specialized pollination systems, which is obviously a bias in my data set. Pollinator shifts are more frequent in certain regions, such as Australasia and Southern Africa. The traits that have been traditionally associated with pollinator-driven speciation, such as nectar spurs or zygomorphic flowers, appear to be not so important. And pollinator shifts are most common within pollination systems, but otherwise are possible in almost any direction. And finally, I'd like to conclude by saying that phylogenetic meta-analyses are a powerful tool to obtain a bigger picture in speciation research. Thank you.
Could have been biased or could have been driven, is perhaps a more correct word,
one thing that I already did in that context is by only choosing one particular study of that kind. The literature is full of studies of fig wasps and fig associations, but I choose to only include one of those studies. So in that sense, because figs are a very specious group, I think it's important that they are represented in the data set. And I did try to minimize the effect by only choosing one study, in a way. That answers your question.
Give us a scenario or two that would suggest why a pollinator shift would happen in nature.
Absolutely. This is basically the Grant-Stebbins model, which I didn't want to go into due to time constraints, but the process-based explanation is basically that there's a geographical mosaic of pollinator species. What happens when a plant disperses out of the range of its native pollinator is that it will adapt to a more efficient pollinator. This is basically what Stebbins in the 70s already suggested. And that is the process that would drive pollinator-driven speciation. And indeed, we find a lot of ecotype examples where this seems to be the case, especially in southern Africa, where there's a long-tongued fly pollination system, which is very patchy. And we do find that different species, or several species, have adapted to long-tongued flies at a very local level.